Hello, my name is Yair Schiff, and I will be presenting our work Caduceus. This is joint work with my wonderful collaborators, Chia-Hsiang, Aaron Gokaslan, Tri Dao, Albert Gu, and Volodymyr Kuleshov. We begin with some motivation for this line of work. Namely, the hypothesis that language modeling is useful for biological sequences, as the pre-training objectives of these models directly lend themselves to learning important concepts, such as conservation and evolutionary pressure, which enables these models to then be useful in downstream task applications. And indeed, recent years have seen success in applications of language models to the protein domain. This has led to recent line of work extending this paradigm to DNA sequencing, with the idea that moving to this domain that contains both regions that code for proteins and non-coding regulatory regions will enable us to understand biological bases and can extend to new tasks such as genome annotation that current protein models cannot solve. However, with these new applications come a new set of challenges. In this work, we focus on the following three unique aspects of DNA language modeling. First, we note that unlike in proteomics, genomic sequences can entail long-range interactions. The tight coiling of DNA means that distal nucleotides up to 1 million base pairs away from a gene can be brought into close physical proximity with a gene and exert regulatory effects. Additionally, unlike in the natural language domain, left-to-right causal modeling may not be sufficient, as base pairs both upstream and downstream of a gene can have interactions, necessitating a bidirectional model. Finally, the unique double helix structure of DNA brings with it a reverse complement symmetry that models should respect, as previous work has demonstrated. Reverse complement symmetry can be explained as follows. In DNA, both strands contain equivalent information, with one strand being the reverse complement of the other. That is, we reverse along the sequence dimension and the channel dimension, flipping A to T and C to G. Then, we desire that the network outputs commute with this operation, namely that they also flip along the sequence and the channel dimension. That brings us to our introduced Caduceus model. Caduceus implements bidirectionality on top of the long-range Mamba module and incorporates a reverse complement equivariance inductive bias. This enables us to improve performance on downstream tasks even relative to much larger transformer-based DNA language models. And Caduceus is the first model to account for all of these three unique challenges in genomic sequence modeling. We now show how the different design decisions of this model achieve this. First, by building off the recently proposed Mamba module from Gu and Dao 2023, we are able to leverage the long-range capabilities of this operator. In sequence modeling, two ends of the compression extreme can be characterized as transformers and RNNs. While transformers have recently dominated the field owing to their ability for all tokens in a sequence to interact, they are not well suited to long-range inputs due to the quadratic scaling that this entails. In contrast, RNNs scale linearly in the context size and have theoretically infinite contexts, but the limited capacity of their hidden states limits this expressiveness, and they have been historically difficult to train efficiently. However, the recent resurgence of linear RNNs with hardware-efficient implementation has posed a viable challenge to the powerful transformer model. Mamba was able to match performance and scale better than even highly optimized transformer models and training recipes, which led us to use it as the backbone of our model. To render Mamba bidirectional, we run modules twice, once on the forward and once on the reverse of a sequence, employing targeted weight sharing to reduce the memory footprint. We call our memory-efficient module BiMamba. Looking at the Mamba module, we notice that the vast majority of the parameters are contributed from the linear in and out projections. By tying these projection weights and letting the sequence modeling components vary for forward and backwards modeling, we are able to drastically limit the number of parameters added from incorporating bidirectionality. Finally, to achieve RC equivariance, we leverage another parameter sharing technique. Namely, we run a Mamba or BiMamba module on a sequence and its reverse complement, and we concatenate the outputs along the channel dimension, while fully sharing parameters of both of these passes. We dub this module MambaDNA. This approach renders the MambaDNA provably RC equivariant. Putting these components together yields Caduceus, the first of its kind RC equivariant DNA language model that scales efficiently to long-range sequences. Moving to the experimental validation of our model, we first highlight the effect of the different design decisions. First, we see in the blue chart to the left that left-to-right Mamba modules outperform left-to-right hyena DNA, a recently long context model, on pre-training tasks. Additionally, in the middle panel, we see that our strategic weight sharing in BiMamba, which allows us to avoid having to cut the network depth in half over the naive implementation of bidirectionality, also improves on the pre-training tasks. Finally, we see that incorporating RC equivariance improves the pre-training objective as well. On downstream tasks, we also see that Caduceus outperforms other DNA language and supervised models, even those with orders of magnitude more parameters. Notably, on the task of classifying causal EQTL SNPs or mutations, the Caduceus model outperforms hyena DNA in a much larger nucleotide transformer model. It even outperforms a well-regarded and former baseline, especially for sequences where the SNP is greater than 100,000 base pairs from the transcription start site, which presumably are sequences that require longer-range sequence modeling. To summarize, in this work, we introduced bidirectional and RC equivariant extensions of the Mamba module. We used these components to build Caduceus, a novel DNA foundation model, and showed how this model can improve over previous work on a range of downstream applications. Thank you for your interest in our work. To learn more about it, please stop by our poster session or check out the paper and code listed here.